What you see here is the structure of an antibody, and I'm going to talk about antibody structure and function. Okay, so here is a picture of an antibody, and I'm going to start labeling the whole thing now, and that's a little bit difficult to see now, white on white. It doesn't matter. This top part over here, this, you see that the antibodies are Y-shaped, right? And this top part over here is known as the variable region. And this part down here, that is the constant region. And you see that the variable region contains here, that's difficult to see, okay, that is the antigen binding site. Binding site. And you see that an antibody has two antigen binding sites, one over here, okay, one over here, yeah, and on here on the other side. So these are the two antigen binding sites here at the tip. So okay. So that is one way of classifying that, of describing, but there is also something called a light chain and a heavy chain. So these long ones over here, these long chains, a little bit difficult here to see. Now that is called the heavy chain. Okay, so there are two heavy chains, and this one over here, have a problem. Don't ever do this on an exam, labeling it like this, okay? It's a light chain. So an antibody or immunoglobulin is made of 4 polypeptide chains. So now you can guess, where are they made in the cell? Well, which part of the cell makes polypeptides? Well, it's the ribosome, of course, okay? So that is the thing, and that's how they look like. In reality, they're not just straight, but they're actually intertwined quite a bit, and it's a globular protein, and this means it's round and kind of compact and circular a little bit. Spherical would be probably the better one. And what you see is that the antigen binding sites, these are always the ones that are different because they have to bind against different antigens. And you can see that the antigen here and the antigen binding site fit together like a lock and a key, okay? And therefore they attach to each other, and therefore the antigen can be deactivated. So now let's see how this actually can work, and I'm going to move on. How do antibodies actually attack or neutralize? How do antibodies neutralize antigens? So first of all, they work in one way. Could be that they block the binding of viruses. So if I have a cell over here, okay, let's say that's a body cell, okay, and on this body cell I have my body own antigens, right? And they do whatever they're supposed to do. I mean, there are thousands of different functions that surface proteins can do. And over here I have a virus, and this virus would like to attach to this cell. So the virus looks like this, right? And what it wants to do, the virus has now over here antigens, AG, I just called this. Okay, these are the virus antigens here, and these are my own body own antigens. Okay, and why does the virus have these antigens? Because it wants to attach now to my cell antigens and to stick to them. So I'm just going to draw this over here like this. So that's how it wants to, how it's going to look like. I'm going to draw the virus here again, right? Yeah. And this allows the virus to be taken up into the cell, right? And then it starts to copy itself inside the cell. But if I have now antibodies, and these antibodies bind against the antigen of the virus like that, if they do that, I mean, the antibodies are probably drawn a little bit too large, but I hope you get the point, then it is not possible for them to bind against the cell, okay? And that's how by blocking the antigens of the virus virus cannot enter the cell anymore, okay? So that is that's number one, okay? How antibodies can work. And the second one is antigens can be cross-linked. Now what does this mean? Now my antigens are little circles over here, okay? That's my antigen, and I'm adding the antibodies, and they cross-link. They cross-link like this, and therefore they they're not soluble anymore. Okay, it's this cross-linking. I hope you get the idea. Yeah? And therefore the antigens are not soluble anymore, and now we have this clot of antigens floating around, which can then be removed by other cells, by phagocytes and so on. Okay, so that's a neutralization by cross-linking antigens. So the next thing, so you can inactivate toxins, and that's a little bit very similar, I would say, probably to the cross-linking. No, but toxins are actually much smaller. Okay, so let's say there are certain toxins. Okay, and what can happen now is that the antibodies bind against these toxins and deactivate them. That's kind of straightforward. Okay, and the last thing is antibodies can make holes in bacteria, and this is known as the complement system. You don't have to know this word, but this is a very well studied and intensively studied thing. What you have is you have a bacterium over here, and the first thing is that there are antibodies binding against the bacteria. Again, I'm drawing this way too big here, okay? And this causes the formation of a hole in the bacterium. So there are other proteins that arrange around the antibody to make a hole in the bacterium, okay? And it's and then the contents flows out and the bacterium dies. That's supposed to be an arrow with the cell contents flowing out. Okay, so it starts. Well, the antibodies themselves don't make the holes, but they start making holes, holes in bacteria, and this kills them. Okay, kills. How do you spell kills? Kills them. Okay, the bacteria. Okay, so these are a couple of ways how antibodies work, and it could be like this, that for example, if you're bitten by a snake and you go to the doctor, they will give you so-called an anti-serum, which contains antibodies which inactivate the toxins of the snake bite. Okay, so that is it. Okay, that's it.